gland is a specialized group of cells that synthesize, secrete substance and hormones. Now, it's a generalized description of the glands. Now, where hormone is coming, that is the gland is a specialized group of cells that synthesizes and secretes substances and hormones. Now, hormones is secreted by the endocrine glands. Okay. Now, glands are derived from epithelium. It is derived from the epithelium because the epithelial tissue, they are modified and they come and to relate themselves into the the function they're destined to be performing. Because see, all cells, if you see your embryology is obviously cell is taking. So the cells are, each cells are destined to do some function. Now, mucus is a noun and mucus is an adjective. Now, for example, mucous cells secrete mucus. Now, you just see this embryological stage, pre-bud stage, surface epithelium. Initial bud stage, the pseudo-glandular stage, see. Now, canicular stage, the glands and terminal bud stage. Now, a portion of this, if you take an embryological tissue from the fetus, then you will be seeing these in stages of what stage the embryo is in. Okay. Now, whole embryo, you would not going to do histology to detect the gland. But now, see a portion, like endoscopically, if there is a malignancy in the stomach, then the endoscope, it takes bits of tissue. So, it can be just a small piece, some small piece. Now, that will be prepared for histopathology and you can view it under the microscope. You will be seeing all like an inch when you see under the microscope. It is inch and there is numerous cells that you are already viewing already. The epithelial cells you have viewed. Now, a portion of the tissue is taken. And what you see is this picture. This is the striated duct, intercalated duct, myoepithelial cells, serous, Demilunes, serous cells, mucous cells. Now, myoepithelial cells, it has to contract and secrete. Myoepithelial, an element of muscle that has to contract and goes into the teeth. Now, you see the surface epithelium. This is the capillaries, bud stage. This is all embryology, development of the endocrine gland. Now, classification of glands are based on number of cells. Unicellular glands, they are single cell that perform secretory function. Multicellular glands, they have groups of cells based on presence of a duct. Exocrine gland, they use duct to convey their secretion. Endocrine glands, they do not have ducts and secrete secretion directly into the blood. Now, this becomes your endocrine and the exocrine gland. Now, based on the manner of secretion. Now, Merocrine glands, they secrete by exocytosis. Apocrine glands, they secrete by shedding of apical portion of the gland. Holocrine glands, they secrete by disintegration of entire cells. So, apocrine, merocrine, holocrine. It can come as a small note or it can come in MCQ. Okay. So, plus what you know, you have the basics. So, you have to know this, basics. What is the merocrine gland or apocrine gland and holocrine glands? Now, classification of glands based on number of cells, unicellular or multicellular. Okay. Presence of duct, exocrine with duct, endocrine without duct. You can quickly take a note of this. You have it in your book, please don't, you take your histology. You have brought your histology textbook. Why? I told you already, kya, kitna class ho gaya. See, if you disobey, I have nothing to do. I cannot punish you and I won't punish you because this is not the place for all these things. And if you are negligent, you will realize after a mock test will be given. Any day can be given. So you will realize what I am telling or Abhijit sir is saying, or I might be talking a bit more. Faltu kotha onek kichu boli ami. It might be faltu for you, it might not be faltu for him, or it might not be faltu for me, because I have no right to waste your time. Now, what I want to say is that you see, now you take it, you bring your books every day. Now, see the surface epithelium, duct secretive was not found in adult, presence in embryonic period during development of glands. Okay, now you see the sebaceous gland. This is the skin. Hair is hair follicles, and these are the sebaceous glands. Now, this will be for long questions. Now, what we'll go to the histology lab and get things in place. Now, see, this is a sebaceous gland. These are sebaceous acini, hair follicle, branched acini, and branches of acinus. Now, the goblet cells, simple tubular cells, appendix. This is the appendix. Okay. Merocrine sweat glands, pancreatic acini, pancreas, exocrine part. Now, let us now, classification based on nature of secretion. Serous glands secrete protein-rich watery secretion. Serous glands have several serous acini on secretory unit. Serous acini are lined by pyramidal shaped simple columnar epithelium. Identification features: round basal nucleus located at the basal lamina, large amount of rough endoplasmic reticulum. Protein synthesis lies in basal regions. It gives basal basophilia stained in with hematoxylin. Well developed Golgi complex. Now, mucous glands secrete thick mucoid secretion. Mucous glands as mucous acini as secretory unit. Mucous acini are lined by pyramidal shaped simple columnar epithelium. Mucous acini has wider lumen and then serous acini. Example, sublingual salivary gland. This is a sublingual salivary gland. Today we will see identification features: flat elongated basal placed nucleus, empty cytoplasm. Synthesize and store the mucus in the form of mucogen granules. These granules are lost during tissue processing for slight preparation for H&E staining. This is it gets lost with the action of the different grades of alcohol or this thing. You don't see it actually. Okay. Now, this mixed glands which secretes both mucus, mixed gland secretes both mucus and enzyme-rich watery secretion. Mucous acini with serous demulins are secreted. Identification of mucous acini is capped with serous cells that form serous demulins. See, types of acini. Now, pyramidal cells, spherical nucleus at the base, apical secretory granules, narrow lumen, and myoepithelial cells. Now, flat basal nucleus, pale cytoplasm, white lumen, and myoepithelial cells. This is the serous demulins. Mucous acini with serous demulins. Mucous acini. Serous acinus. Now, submandibular gland. See the picture of the submandibular gland. Mucous acini, serous acini, mucous acini with serous demulins and serous demulins. This is the submandibular gland. Now, difference between mucous cells and serous cells, that you read more of theory. Difference between serous and mucous cells. Merocrine, Holocrine glands, this is the picture. We get in here all histology classification glands on mode of secretion, Apocrine. Now we'll go, I'll arrange the slides there and I'll call you. Parotid is the tongue. Okay, it has numerous mucous glands. And the buccal cavity, lips, your oropharynx, then your palate, soft palate, pharynx, all these have, because what is the main function of the salivary gland? You have to know that it transports the food and also it helps in enzymes and digestion, and it keeps the tongue moist. Now, what happens at times, you say you go on speaking or this thing are dehydrated, you cannot move the tongue properly. তুমি নিজেও কথা বলছো, অনেকক্ষণ জল খাওনি, you have not taken water, and these watches, what are smart watches, take a sip, they give you a signal, but don't depend on the signal, you go keep on drinking. There are 4 slides, it is from the same block, you just go and see and correlate. 10 of you come, 10 of you come, and 10 of you come. There are 4, there means each slide, there will be microscope, 4 microscopes are there. It is from the same tissue block, See, this is the, I told you the block, like this, the wax mold. Now this is the tissue. These all 4 slides are prepared from the same block. We won't bring any new slides or something, or that examination, we don't do it. Here's one book. You please find out. Okay, and here's one book. এখান থেকে দেখে নাও. You just correlate with the pictures. খাতা বার করো. The viscera. Then you know the tissues, the connective tissues, how it is molding. Because see, it's the gland will be small. You see the pattern that I showed you. You are seeing similar here. Now you can see that. I'll just show you. You please see and see the microscope. Okay, see the microscope. Bring your own histology books for next day. Bring your own. Hey, Solo, I told you it's a binocular microscope. Don't see like this. You open your, if you feel uncomfortable, you have to make a habit. Because those days in school, you didn't get this thing. Now you see like this. You rest, now it's beautiful. Now you see the pattern, how it is there. Now you see I have focused it. Now it might be that you have to adjust a bit of this thing. You can bring your this thing, if your bridge of the nose is, or you have a white face, then you can accordingly you see like this. Hey. প্রথম দিন বলেছি you have to not like this. একটা দিয়ে দেখবে কেন? You having binoculars microscope, দুটো চোখ। না তুমি দেখো এইরকম। See, this একটু অ্যাডজাস্ট করো, এই দেখো এই কোর্স Now this is the fine adjustment. Fine adjustment. Now accordingly you look into it. A lot like this. A lot like this. তুমি তো দাঁড়াও একটু দাঁড়াও Now what you are seeing, you can see like this. You can see like this. You can see into this. Do to thumb. Chalo. Now you can see this 180. 180. You can see like this. Now what is this? If you see it in the microscope like this, it too. Then your vision go to the retina and on this. Then your. So you see that is a school microscope. Now you are having a binocular microscope. You adjust like this, see this gland, draw the figure. ঠিক আছে না এখানে দেখো এটা This is parotid. This is the parotid. Okay. Now these are the connective tissue. That if the yellow part, this part, it has to be in place. What structures will give a support? Now these things are penetrating inside, and each lobule is formed. Now, what you will be seeing in the books is that one stroma, the connective tissue, and that is supporting the other. When you take a section, you will be getting that connective tissue picture. It has to supply with blood vessels. You will be seeing arteries, and the thickness of the arteries more than the vein. Now you can see small arteries there. You take just 10 minutes, observe, see the book, see the illustration, and this thing. This, if I take a tissue, parotid, say if there's a parotid adenoma or some nodule, a patient comes, it is there. Now you take a biopsy. Biopsy means you first do a needle aspiration cytology, FNAC. Fine needle aspiration cytology. You just don't cut immediately. Now you get a confirming report. You might not get a confirmatory because you don't know exactly in which place that tumor cells are there. But you get a report. Of course, most of it you get that it is a non-malignant, benign, or malignant. Okay. Now, the idea is that you can small bit of tissue is there. In between, connective tissue is there. The acinar cells are there. The mucus secreting cells are there. My epithelial cells, and it is secreting, and then it goes into the ducts, and that duct opens into the crown of the second molar tooth. Now, what is I want to stress is that you have a conceptual in the gross, and then you see in the picture that is drawn by hand, illustrated. Then you go, you view this slide that is the histology picture in the now you see, this is handwritten drawn. এটা আমার বইটা দাও তো histology book. See the parotid I showed you, parotid gland. Now it is like this, the picture. What you are seeing is this submandibular gland. See all these connective tissue, adipose, some fat tissue is there also. Similarly, see here. This you have to draw in your book. Main thing, you have to draw this in your book. You have to be acquainted with the slides, with your whatever is there in the slide. That what you do is that when you are viewing, you just draw a circle, and then you place. Just you don't take hours in the class, but you just level. It becomes easy to retain and also to reproduce. দেখেন আরেকবার, lots of time is there. আর তিন ব্যাচ আর দু ব্যাচ আছে ওখানে, দেখে নি ওখানে মাইক্রোস্কোপ আছে, দেখে নি। এটা তো এই দেখো, এই দেখো, দুটো চোখ দিয়ে দেখবে, দুটো চোখ, view with two eyes, view with two eyes. Straight.
একটা ব্লকেরই চারটা স্লাইড, তো খুব একটা তো বেশি ডিফারেন্স হবে না একটা স্লাইডের মধ্যে, মানে মাইক্রোস্কোপ আমরা দেখতে
এটা
আচ্ছা স্লাইডগুলোর মধ্যে
দেখি আমরা নতুন পেন, হ্যাঁ মানে বেসিক, মানে what you have to do is seeing this salivary glands, you go back. That's why I'm telling you, bring your histology textbook here, sit for half an hour. After viewing, you see it, sit in the over here and go back to demo room and see the pictures. You listen to me, otherwise you will be in chaos. What I'm trying to say is that you just draw the rough picture like this. Now this is one duct. Now these are the lobules. This is the lobule. This is the big lobule. Huh? This is the big. Like this, you draw like this, rough. This is a big lobule. This is a big lobule. In between, the acini cells are there. Now you draw like this. You draw the basal epithelial cells. The basal epithelial cells. This is basal epithelial cells. Now basal epithelial cells. Like this. এখানে সেই একবারে ফাইন করে আমি আঁকতে বলছি না। What I want to do is that you have to convey the message. In the exam, you are not asked to draw. You are asked to identify. So, if you can reproduce, means from the first, as you observe, you draw a sketch, you see the histology sketch, plus you are seeing the slides. I'm making it simple for you. And when you are seeing the microscope, binocular, দুটো চোখ ব্যবহার করো। চশমা নিয়ে দেখো, অনেকে দেখে, অনেকে চশমা খুলে দেখে। And there are some adjustments, it is fine adjustment. But don't move the stage. In the examination, you don't move the stage. Stage micrometer তুমি হাত দেবে না, কারণ তোমার only like this, just like that. Now you see, this is the stage that is fixed by the examiner. Now, in the class, you will be moving and seeing a whole topography. See, this is a picture. Who have been to Lakshadweep, Maldives Islands? See, you compare with the islands, you relate. যখনই তুমি একটা Lakshadweep, beautiful picture you're seeing in your Facebook and this thing. Now, when you are seeing, you see that relate in the histology. Different small islands. The islands are having cottages. These are the acinas. This is the lobules, separated over here by connective tissue. And each tissue has to be supplied by some blood vessels for its nutrients and this thing, actions and all. Plus carrying deoxygenated blood, oxygenated blood. See, these are machinery. These are machinery. So it needs oil. Now, regarding the salivary gland, that is the 10% in tongue, born Ebner's glands, and also the oral cavity, whatever it is. Now, 90% is by the parotid, sublingual salivary gland, and submandibular salivary gland. So sometimes the salivary glands are here, sometimes there is a calculi formation in the duct, salivary gland, and there, the tongue, the papilla are there. It goes salivary, submandibular glands open there. That's why there might be a question in your scilogram. What is a scilogram? It's a special dye you inject and then take a X-ray radiograph to see if it is a blockage due to some tumor or something, or it might be due to some calculus. Okay.